Enzymes, relatively large molecules of protein, are biological molecules that catalase or speed up chemical reactions. Every cell makes hundreds of different enzymes to carry out the reactions necessary for life. The molecule or molecules on which an enzyme acts is called its substrate. Enzymes recognize only one substrate or a few closely related substrates and convert it into specific product. Each enzyme is specific because it is folded into a particular three-dimensional shape. Within the folds of each enzyme is the active site, the place where the substrate fits and where the chemical reaction takes place. Enzymes work very quickly, often catalyzing thousands of reactions per minute. The rate at which an enzyme works is influenced by many factors including temperature and pH. Enzymes have a temperature and pH at which they work best, and if an enzyme is exposed to extremes of heat or pH, it won't work at all. The interactions that hold the protein in its particular shape become disrupted under these conditions, and the three-dimensional structure unfolds. In this case, the enzyme is said to be denatured. The enzyme studied in this experiment is called catalase. Its job is to break down its substrate, hydrogen peroxide, or H2O2, which is a naturally occurring poison. Without catalase, H2O2 would kill the cell. The products remaining after catalase does its job are oxygen gas and water, two very non-poisonous molecules. The dependent variable of the experiment was the reaction rate, and the independent variable of the experiment was the different temperatures of the catalase. The first control was water dipped chads in 3% H2O2. The second control was dry chads into H2O, and the third control was chads soaked in boiled potato catalase. The constants of the experiment were the size of the chads, the time soaked in the enzyme, and the substrate concentration. The hypothesis was that if the chad was soaked in cold potato enzyme, then the reaction rate would decrease compared to that of the room temperature reaction rate. If the chad is soaked in hot potato enzyme, then the chad will not rise at all.
Materials include 6 beakers, filter discs, safety goggles, gloves, 3% solution of hydrogen peroxide, a thermometer, tweezers, catalase, pipette, ice, a microwave, a graduated cylinder, and water. The first control was set up with a 3% hydrogen peroxide solution and water-dipped chads. The second control was with water and dry chads. The last control was with a 3% hydrogen peroxide solution and boiled potato catalase-dipped chads. The independent variables were then set up. The cold was set to 18 degrees Celsius, the room temperature was set to 28 degrees Celsius, and the hot was set to 38 degrees Celsius. In the first control, water-dipped chads were dropped into a 3% hydrogen peroxide solution. The reaction rates were then recorded. In the second control, dry chads were dropped into water. The reaction rates were recorded once again. In the last control, boiled potato catalase-dipped chads were dropped into a 3% hydrogen peroxide solution. The reaction rates were recorded once again. 3 chads were dipped into room temperature or 28 degrees Celsius catalase enzyme for 15 seconds. The chads were dropped into a 3% hydrogen peroxide solution. The reaction rate was timed and recorded. Three chads were dipped into cold temperature or 18 degrees Celsius catalase enzymes for 15 seconds. The chads were dropped into a 3% hydrogen peroxide solution. The reaction rate was timed and recorded. Three chads were dipped into hot temperature or 38 degrees Celsius catalase enzymes for 15 seconds. The chads were dropped into a 3% hydrogen peroxide solution. The reaction rate was timed and recorded.
The purpose of this experiment was to determine how different factors may affect enzyme function. The lab was conducted in order to identify relations to enzyme activity within human cells. The null hypothesis of this experiment was that all three enzyme temperatures would have the same rate of reaction. The null hypothesis was rejected because the chi-square value of 19.6 was larger than the critical value of 5.99 for the 3 degrees of freedom. The results showed that all of the enzyme-dipped chads floated in the H2O2 solution except for the enzyme that was heated to 38 degrees Celsius. One error in this experiment was the effect that the room temperature had on the temperature of the enzyme being tested. An improvement for this error would be to conduct the experiment in a room with the same temperature as the enzyme being tested. This would ensure that the temperature of the enzyme would not increase or decrease. The overall hypothesis of this experiment was that if the chad was soaked in cold potato enzyme, then the reaction rate would decrease compared to that of the room temperature reaction rate. If the chad was soaked in hot potato enzyme, then the chad would not rise at all. The hypothesis was accepted because when the temperature of the enzyme was increased, it denatured, and the chad did not rise. When the temperature of the enzyme was decreased, the reaction rate decreased as well, because the enzyme was not placed in its preferred environment. The overall findings for this experiment showed that the enzyme worked best in room temperature.
Here we celebrated with celebratory red nail polish. As you can see, Riley Jordan Troyer displays her lovely blood red nails for us.
It's crimson.
Did it evaporate?
Evaporate. No way. Are we supposed to put it in there? She told us to microwave. Apparently that was too long. Oh my god.
Let's go.
Is there anything in there?
It's dry AF. Look.